In this session, we will talk about the T-cells formation and basic structure of T-cell receptor. The process of formation of T and B lymphocyte is called as lymphopoiesis. Lympho means lymphocytes, which are T and B cells, and poiesis means formation. In bone marrow, the hematopoietin stem cells matured into common lymphoid progenitor cell. It can become B cell or T cell. But in this video, we will only talk about the T cells. So, to become a T cell, it moves to thymus and converts to thymocyte, and then matures to T cell. As we have discussed in our previous video on major histocompatibility complex, that cytotoxic T cell has T-cell receptor and CD8 co-receptor, and MCH-I has groove-presenting antigen that binds the T-cell receptor, while CD8 co-receptor binds the Beta-2 Microglobulin of MCH-I. Same pattern is followed by T helper cells. These cells has T-cell receptor that binds with the groove of MCH-II presenting antigen, and CD4 co-receptor that binds the Beta-2 Microglobulin of MCH-II. But have you ever thought that, what if receptors of T cells do not possess a perfect and complete structure? Obviously, it won't be able to perform its function, and cannot recognize and binds to foreign substances. So what's about the immunity that we acquired? So it means structure of T cell is very important. Let's understand its structure together. There are two types of dimers. Heterodimers means two different monomers, such as alpha and beta. But homodimers means two similar monomer, such as alpha and alpha. T-cell receptors are heterodimers. What does heterodimers mean? As we have discussed, that heterodimer means two different types of monomers. These monomers are alpha and beta monomers, while other are gamma and delta monomers. These monomers make T-cell receptor. So one type of T-cell receptor can have alpha and beta monomers, while other types of T-cell receptor can have gamma and delta monomers. Mostly, T-cell receptors can have more alpha and beta monomers, rather than gamma and delta. Have a look of these monomers. Gamma and delta T cells are rare in humans, but has a major proportion of T cells in other animals, including cows, pigs, and sheep. T-cell receptor with alpha monomer, which has molecular weight of 50,000, and beta monomer, which has molecular weight of 45,000, are more common than the T-cell receptors having gamma and delta monomers. As antibodies have constant region and variable region. Constant region is same in same class, but variable region varies from one class of antibody to another class of antibody. Through this variable region, antibodies are able to detect different antigens. Similarly, T-cell receptor has variable region and constant region. As it is the constant portion of alpha chain, so it is represented as C-alpha, while this is the constant portion of beta chain, so it is represented as C-beta. Now, this is the variable region or domain of alpha chain, so it is represented as V-alpha. This is the variable region or domain of beta chain, so it is represented as V-beta. This variable region helps the T cell to detect antigen, presenting the major histocompatibility complex to T-cell receptor. There are disulfide linkages between the chains, so they are called as interdisulfide linkage. They are called disulfide due to presence of two sulfur atoms, while inter means between the two chains. These disulfide bonds are also present within the chain, called as intradisulfide linkage. intrameans within the chain. These linkage are in variable region and as well as in constant region that will be discussed later in this video. This is the basic concept. In our next session, we will discuss the structures of T-cell receptor.